E. coli. We were talking about the fact that when we're born, we're colonized within 48 hours, which is pretty amazing to get our bacteria so quickly. And but for the most part, they tend to be benign and they help us. So E. coli is actually a very helpful organism. E. coli is considered to be a coliform, and what that means again is it's going to digest glucose and lactose. And in particular, E. coli is considered to be what's called a fecal coliform. And fecal coliforms are ones that are associated with the intestines of mammals. So we are animals, we're mammals, and so we have E. coli in our fecal matter. What that means then is if we find E. coli in water, that means that fecal matter has come into contact with the water. And that can, of course, affect water quality. So we have to be really careful with how we, treat water that has fecal deposits in it. Now, the reason we worry about this is because exposure to water with feces, it could result in an infection. Now, the reason we say it doesn't always result in an infection is because, remember, I'm putting a little bit of fecal matter in a huge amount of water, which means the bacteria is going to be very dilute. So I have to ingest enough of it to actually cause an infection. So the idea behind this then is that the fecal coliforms can be present, but they might not always cause disease. And the reason they might not is because as I'm drinking the water, there's no guarantee I'm getting enough bacteria to actually make me sick. So it's kind of like a game of Russian roulette. I've had people tell me, well, I hike all the time and I drink from the streams and I never get sick. And that's fine. It's probably because it's so dilute, but they're playing a dangerous game because what happens if they run into water that is full of fecal matter? They might actually get themselves sick from it. Again, if you end up with diarrhea from drinking contaminated water, the best way to handle it is to, first of all, either boil the water to avoid any contamination. So if you travel, you can boil water. That'll get rid of any bacteria. Then what you can do is if you're having diarrhea, add either sugar or salt to the water. So a little bit of sugar, a little bit of salt, and that will help restore your electrolytes. Because one thing we don't want to have is we don't want to have you get too dehydrated. So this is like one of the things that we teach, people that are traveling nurses or traveling doctors, is these are quick electrolyte solutions: salt, sugar, water. You can get people to feel better pretty fast. So how do we test for coliforms? Well, one of the ways we want to test is we want to do something where we're taking a lot of the water sample. The idea behind this is we might luck out and hit the microbes that are present. Again, since they're so dilute, it's not always easy to find them. So we use something called a most probable number test, which is also referred to as an MPN. That's the acronym for it, most probable number. And what this is it's a tube of broth that is going to have lactose in it. It's going to have a Durham tube, so we can look for gas formation. And we're going to incubate these tubes and see if we see any growth. And if we do, then that's going to indicate that we can have coliforms present. And we can also have special reagents in there that can even indicate if we have fecal coliforms present. Another thing we can do is we'll do membrane filtration. So if I'm working at the water company, I'll set up a bunch of MPN tests. So do a large sample, set up a bunch of test tubes with water samples. And then I'll also do a membrane filtration, which is basically where I have a filter paper and I'm pouring in several gallons of water. So again, the idea is I'm trying to get a big enough sample to find anything that's dilute in there. As I pour in those gallons of water, the water will run through my filter and my filter will get caught with bacteria. I can then pull the filter off and put it onto a petri dish and any bacteria that are present will grow. Depending on how they look, I can tell what kind of contaminants I have. So for example, if I have a general bacterium, which is just eating glucose, it'll produce kind of a light pink. If I have one that can eat lactose, which means it's a coliform, I'll get a purple color. If I have one that is a metallic sheen, then that indicates that not only am I having a coliform, but I have a fecal coliform. So we saw that a little bit in our labs, right, with the sprouts. We saw a few that were fecal coliforms. Here's a picture of one of those filter papers, so you can see the lines. The filter paper is really thin. You just peel it off of the filter apparatus, put it right on top of the petri dish. And the media can diffuse through that filter paper, and then we can grow our bacteria. And then based on that, we can sample several gallons of water and hopefully catch any bacteria that are present. Now the MPN test is another, yeah, Brianna.
The endotype is the one that we use on the last experiment, right?
Yes, the beak. Yep, you got it. Now, the nice thing about our MPN test, and we do this a lot with water sampling, is it's a quick and statistical way to determine how many organisms are present. So the idea behind this is we're going to set up several sets of tubes. And you can use 3 tubes, 4 tubes, 5 tubes, depending on what setting you're wanting to follow. I'll show you a table that explains that. When you set them up, we're going to have some tubes that test for 10 mils, some that test for 1 mil, and some that test for 0.1 mils. They're set up in a series though. And again, the idea is we're just doing broad fishing. We're trying to see how many microbes we can find with as much of a sample as possible. So that's why we're setting up different amounts. We'll grow the organisms for 48 hours, and we're going to look to see if the media changes color. When we first start, the media is going to be red and clear. If my organism can eat glucose, it'll turn kind of a cloudy red. If it can eat glucose and lactose, it'll turn a bright yellow. I'm also looking at these Durham tubes that are inside because remember E. coli produces gas. So if I see gas, then that can indicate I have a fecal coliform. So it's like a big, easy way for me to tell what's going on. So with this then, my coliforms are going to be gas production. My fecals are going to be gas production, as well as having a special component in them that'll make them fluoresce under UV light. It turns out that fecal coliforms have a special enzyme associated with them. And I can use this enzyme under UV light to make the tubes glow if there's fecal coliforms present. I'll show you a picture of that in just a second. So the idea behind this is we set them up, we read them out the next day. If they're this purplish red color, that means they're negative. If they're yellow, that means they're positive. We count the number of positives. So here I have 4 out of 5. Here I have 2 out of 5. Here I have 1 out of 5. And I compare it with a statistical table. And this table will tell me, based on these results, how many bacteria I have per mil or per gallon or whatever arrangement I'm using in my water samples. So it's all statistical based. Here's a picture of fecal coliform under that UV light that I mentioned. So again, our fecals a lot of times will have unique enzymes associated with them, which we can add to this tube. And it's kind of like when we did our chromogens. If they can break that chromogen apart, they'll fluoresce. The same idea here. If I'm a fecal coliform, I have the enzyme that will allow me to fluoresce under UV light. So if you're in a water lab, the first thing you read out is color change and gas production, and then you go into a dark room and you do a UV readout. So although my friend one time I walked in, she was doing water testing and she was under her desk because she didn't want to go in the dark room, so she just had the UV light under her desk, which was cracking me up. I'm like, what do you do? Like I walked in there and she's like hiding under her desk. I'm like, what are you doing? She's like, water samples. I'm like, okay. Yeah, and it was dark under there. That's why she was hiding under there. So you can do it however you want, wherever you can see UV light. Now, this is a great test to tell us that we have coliforms, to tell us that we have fecal coliforms. What this doesn't tell us is if we have pathogens. We use these to assume that we do. If I see a positive, then I'm going to treat it as if I do. But I still don't know if there's Shigella in there, if there's Salmonella in there, if there's Vibrio in there. I don't know. We're just using these as our indicators. Now, as I mentioned, we can also do that membrane filtration test, but this is one that sometimes isn't preferred because of the fact that it can have issues. If my water is silty, the dirt can clog the filters. And so now I'm seeing more dirt on my filter than bacteria. So I can get interference, is what I call it. So I have to make sure it's a nice, water sample that doesn't have any dirt or debris in it. Because if you're filtering, that's going to show up on your filter paper. The nice thing about this one is only the living cells will grow. So we can really tell whether or not the population is robust because only the living ones are going to be able to survive. Now, I mentioned before that our coliforms are able to use enzymes that allow them to fluoresce. So a couple of different enzymes can be used. First of all, I said that to be a coliform, you have to be able to eat lactose. Lactose is broken down by a special enzyme called beta-galactosidase. And that's the enzyme that helps them eat lactose, so they can eat that sugar. Now, my fecals have an additional enzyme. So they have beta-galactosidase, but they also have something called beta-glucuronidase. That's the one that allows them to fluoresce. So that's the one I'm looking for when I'm looking for a fecal. So I'm looking for beta-glucuronidase. That tells me that they're actually able to fluoresce. Now, I can also do a presence-absence test. This is very similar to MPN, but instead of relying on growth and 48 hours and waiting for incubation in tubes, this test is specifically for that galactosidase and glucuronidase. I'm looking for specific enzymes. And when I'm looking for enzymes, I can tell the test will work faster because I'm not having to wait for them to grow. I'm just seeing if they have the enzymes present. So what that means then is when I set up samples like this, that are the presence-absence, I put in my water sample, and within 16 hours, or sometimes even sooner, I can tell if I have coliforms present by growth, and under UV light, I can tell if I have fecal coliforms, because I'm targeting an enzyme production more so than having to wait to target for growth. Again, it's like the chromogens. I'm looking to see if they're able to break apart components, just like what we talked about with the chromogens. If they can, I don't have to wait for the cells to grow to a certain number to break them apart. Any cells that are present can break them. So I can get reactions very quickly. That's why these guys have a faster turnaround. Brianna, you had your hand up.
It was about the last slide. I was wondering, so fecal coliforms have both of those
Correct.
enzymes?
Yes, that's correct.
Okay.
Because they have to be able to break down lactose too.
Okay.
You got it. So this is kind of a quick and easy way for us to also set things up, and we don't have to set up as many tubes. So a lot of water plants are starting to go this route. The only problem is this does cost more because trying to target enzymes is a little more expensive. So we still have a few labs that are doing like the MPN tests. So what does the regulation state about our testing? Well, there are certain numbers of coliforms allowed in different types of water. So for example, when it comes to drinking water, there are no coliforms allowed. So, we don't want to have anybody drinking coliforms. If you're in a swimming pool and if you're doing a test, you can have growth of 3 coliforms, so 3 colonies for every 100 mils. So the idea behind that is swimming pools aren't sterile. You might have something in them, but as long as they're under a certain amount, they won't cause disease. Recreational water, so that would be something like a water park, can have about 100 to 200 fecal coliforms. So again, we kind of are like, ew, we want 0. But again, that's hard to really always have 0 when you have so many people coming in and out of the water. Fish and wildlife habitat, that would be like Lopez Lake. They have an average of about 5,000 coliforms per 100 mils. And then shellfish, when we test them for like red tide and when we're testing them to see what kind of fecals they have, they're allowed to have 14 fecal coliforms per 100 mils. Aren't those swan boats really cool? I'm going to tell you right now, never go on a swan boat with your child. Because here's the deal, there's nothing to distribute your weight, and you always weigh more than your kids. So you get on the swan boat and your boat goes like this, and then when you're trying to paddle, your paddle's not in the water on their side, so you just kind of do circles. So they look majestic, but make sure you go with somebody who's the same size as you. I'm just going to tell you that right now from personal experience, okay? Because my kid and I were out there just doing donuts on the lake. It was not this exotic experience that I was expecting. All right? Like these little feet were out of the water. You know, it's like I'm trying to lean over. Yeah. And it makes you feel really like you're like huge. It's just not a very good situation. Okay. Wastewater is another one we're going to talk about now. So did that make sense how all those tests work? Okay, so that's for drinking water. Let's talk about wastewater. Wastewater is, of course, our sewage treatment water. And this is where we have our toilet water, our gray water, which is like washing machine and sink water, and all of our gutter runoffs and street runoffs. So we actually have to test sewage water as well because we want to make sure that after we've treated it, it's clear and free of microbes. This poor person right here is having a horrible day. They're in dirty sewage water trying to clear out tree branches that washed in Napa storm. So, if you're ever having a bad day, just picture this person's life, okay? They get to go out in the sewage water and clean out tree branches. So it makes you kind of appreciate where you're at, right? If you don't have to do that. The wastewater process is very easy to do because of bacteria. And it's separated into three main components. There's the primary treatment, where we're going to have a first mixture of bacteria come in. There's the secondary treatment, where we allow more anaerobes to break down fecal deposits. And then there's the tertiary treatment, which is where we actually release the water. And you're going to be amazed at how clean the water is when it gets released. So with the primary treatment, the first thing we're doing is we're removing everything that shouldn't be there. Sticks, tampons, garbage, large materials, anything that's been flushed or put into the sewage pipes that shouldn't be there. Those are pulled out with nets or whatever we need to use, and then they're thrown in the landfill. Then the water is moved to what we call a settling tank. A settling tank is just a tank full of bacteria. And what they do is they just keep this tank constantly being filled so the bacterial populations that are present are able to have a constant incoming food supply. So once they get established, now we have these bacteria coming in. This is similar to what they sell, for example, with septic tanks and with trailers. They have special little enzymes that they sell that help break down sewage. Those enzymes are bacteria. You're basically sprinkling bacteria into your septic tanks and into your sewage lines. And what they do is they break down fecal deposits for you. So what's going to happen then with primary is when we're done removing the debris and letting the sewage sit in a tank with bacteria, we use up about 50% of the nutrients that are present in that fecal matter. Now, what that means then is we're starting to set up kind of an anaerobic situation. If I have 50% of my oxygen removed now, the bottom part's going to be more anaerobic, which means for my secondary, I'm going to get more anaerobes. And the anaerobes can really break down fecal matter really fast. I removed about 90% of suspended solids, so that's pretty good with this process because the bacteria are eating them. And at this point in time, if I was to do a quick water check, I would find that about 55% of my water would be fecal coliforms. So I'd find mostly E. coli and Klebsiella, things like that in there. This is what my primary treatment looks like. So as you can see, it's not pretty. I mean, we got some scum and stuff on top, and it's full of bacteria. This is also what is stinky. When you drive by Nipomo on a hot day, I don't know why they have their sewage tanks right by the freeway. Have you guys ever noticed that? They're like right there. And they have their primary septic right there. So that's what you're smelling on a hot day, is the bacteria breaking down the fecal matter. So they're working for you. So now you know as you drive by, you can wave at the bacteria because they're doing their job. The secondary part of our treatment is to move through, allow our anaerobes to break down some more fecal matter. And then what we do is about midway through this process, we move them to a new tank where we have what's called a spinner. It spins around in the tank and it bubbles in oxygen. So we start to reestablish some of our aerobes. Now remember, a lot of our bacteria are going to be where they can go back and forth between anaerobic and aerobic, right? What do we call that again? Excellent, facultative, yeah, facultative anaerobes. So they can go back and forth. So as soon as we pump in more oxygen, we start to stimulate their growth. They grow faster. So now we're increasing their growth. We have the anaerobes at the bottom that are breaking things down. And so now we get this really nice slurry of bacteria that are working together to eat and break down all of that fecal matter. So now, this is what a slurry tank looks like. It has a big spinner. You'll sometimes see them, or a bubble wand. Sometimes you'll see those. There's a sewage plant off of, I think it's Black Road or Brown Road. I always get confused. I think it's Brown Road. It's out near Sheriff Farms. And you can see their bubbles, their bubbling wands with their secondary tanks. Look at the water. Is it looking better? It's looking a little clearer, right? So we're getting that bubbling. We've removed all the large fecal matter. And so now we're getting down towards more bacteria than anything. So now if we went in there and looked at it, we find that about 85 to 90% of the food and oxygen sources have been used up. So now we're putting more back in there. So what that means then is the food, the fecal matter is disappearing, and that's what we want. 90% of the solids have now been removed. And now if I check, look at this count, I'm at 90 to 99% coliforms. So what that means is I've removed all of the harmful bacteria. I'm down to just my coliforms. Okay, and the coliforms are going to continue to keep breaking things down. That leads us to our tertiary treatment. Tertiary treatment is where now I've done my process, I've used my bacteria, and so now the process is I need to get the bacteria out so the water can be released. So with this in mind then, what I'm going to do is I'm going to filter the water. I can filter the water through sand, that's how nature does it, through gravel, but I also will help with additional filtration where I can add chemicals, or one of the easiest things is to do UV light. UV light, we mentioned before, it changes DNA. So if I mutate their DNA, I make it to where the organisms can't live very long. They become unstable. And if I expose them to a lot of UV light, then I really can get rid of them quickly. So here's a tertiary treatment. So this water has gone through a gravel and filter system with gravel and sand. Sorry, I can get that, gravel and sand. And now look at the water as it's getting released. Doesn't that look clear? It's almost like a waterfall, right? And then a lot of times they'll be near like lakes or near streams, and they'll allow the water to slowly release out into nature now. The idea behind this is they have removed all of the bacteria. And again, the way they remove the bacteria is through filtration and through UV light. The UV light are in long tunnels that the water runs through, and there's UV lights all around on the bottom as well as on the top to increase mutation. This is a little bit of an example of water being released. It kind of looks like a waterfall, and here's the end product. What do you guys think? It's pretty awesome, right? So, I mean, this is really clean water. When it gets released, we've removed all of the components. That water gets put back into the environment, and it just basically gets recycled. So this is our sewage processing. So again, pretty cool that we're able to use bacteria to do all of that for us without having to do too much. Okay, so that gets us through all of our water quality. Let's switch gears now and talk about food quality and food testing. So the idea behind food testing is that we really want to make sure our food is safe, that we're able to control microbial growth, and that we keep in mind conditions that can cause contaminants or bacteria to grow. There is an acronym that is used in food micro called FAT-TOM. And what that stands for is food, acidity, temperature, time, oxygen, moisture. These are all factors that affect the growth of bacteria. So these are all things that we have to think about. So, for example, is my food acidic in nature? Think of like tomatoes. Do I need to worry about bacteria with tomatoes? Well, not as much. Yeah, it's acidic, so I don't have to worry about that. I might need to worry about yeast, but not bacteria. Temperature. Is my food being stored at room temp? Is it being stored in the refrigerator? These are all going to affect different risks associated with growing the food and having bacteria growth. So another concept we like to keep track of is something called the hurdles concept. And the hurdles concept says that we do things to prevent growth. So for example, we might increase the acidity in a food. We might have you put food in a refrigerator rather than at room temp. All of these things put hurdles in place that slow the growth down of bacteria or slowing their growth. Now, if one of these hurdles breaks or isn't done properly, then we have to turn to the idea of what's called predictive microbiology. This is where something has gone wrong with our hurdle, and we have to know what kind of impact it's going to have. I'll give you an example. Let's say I work at a store. Okay, we'll say Vaughn's or Albertsons, and they've delivered milk. And maybe I'm short-staffed, and the milk sat out on my loading dock for several hours. Okay, so it wasn't at the right temperature for several hours. I've now removed a hurdle. So now with predictive microbiology, I would call the company and say, this happened, what do you think? And they would say, okay, well, if it was so many hours, we predict we might have this much growth. It should be safe, but pull it from the shelf a week early. Does that make sense? So we have to think about how the food's been processed or handled. And so stores are constantly trying to keep track of this with the food industry. We keep track of it because the hurdles idea is that every barrier we put in place adds up to keep bacteria away. If I take hurdles down, I have an additive effect for growth, meaning that I can start to increase their growth. And if I get the right hurdles in place, they can be synergistic. What does that word mean? Synergy. Did any of you guys know that? Yeah.
They complement one another.
They complement one another. That is perfect. That's a great example. They complement one another. So people that are synergistic, maybe you have somebody who's a mathematician and somebody who's a great English writer, and they work together and they complement each other. Same idea here. My hurdles, my temperature, my acidity, they're complementing each other to keep the bacteria away. So they're antagonistic, meaning that they're not allowing growth. But if I take away a hurdle, then something can happen. So all of these things are in place to keep my food as safe as possible. And again, the reason for this is I want to make sure that I'm not introducing anything that's biological or chemical or anything that's unsafe. And we have random quality controls that are done, that are set in place for every food process that we have, but we also will do random sampling as well. So for example, if I'm working in the poultry line, I'm going to do quality control randomly as I'm preparing the chicken to make sure that the bacteria levels are low. And then once my product is finished, I will randomly pull samples to sample as well to make sure that the bacteria levels stay low. So these all work hand in hand. Even though we're careful though, we do occasionally have things go wrong. We have an estimate of about 46 million illnesses a year based on food. And that can lead to up to about 3,000 deaths a year. And a lot of times these are associated with Salmonella or E. coli 0157 or CBOT. And the issue here is not just the pain and suffering that the patient might go through, but also what their families go through. I mean, if you lose a loved one, you can't replace them. And then also what the company goes through. If they have something that is like, slanderous to them, if there's a company out there that has released contaminated food, it's hard for them to recover. Some companies never do. So there's a lot of cost associated when things go wrong. Most of the bacteria that are going to be responsible for food poisoning are ones that we've heard of before. Salmonella, staph, E. coli, those tend to be the most common. The one at the top, Campylobacter jejuni, this is one that's a little bit rarer. We usually see it with chicken recalls. And Campylobacter can cause a very irritated, almost like irritable bowel syndrome. It causes inflammation in the intestines, and it can result in bloody diarrhea. It's very uncomfortable, and it's one that usually is not always associated with death. As you can see, it has a lower death rate than some of the others, but it's just an uncomfortable infection to have, and it lasts about 5 or 6 days. So you're having a very upset stomach, lots of diarrhea, you're very uncomfortable for those daytimes. Now, with this in mind then, the big four that we're always looking for and we're always testing for are those Campy, the Salmonella, the Clostridium, and the staph. Those tend to be our big four. We do also test for E. coli 0157, which would be kind of the fifth one down here, but thankfully 0157 is pretty rare. We'll find regular E. coli. It is rare to find 0157. So thankfully it's not considered to be one of our big ones. Yeah, Aaron.
Why does E. coli that is not 0157 cause intestinal upset?
Great question. So a lot of times it's because of the number. So we're getting all of a sudden a huge dosage of E. coli. So let's say in our body we usually have around 1 million. And if we eat food that's contaminated, now our number's gone up to 2 million. And it's not our normal flora strain. They're always a little genetically different. So they can be ones that our immune system will recognize as not self.
So they have different antigens on those.
Exactly. And so our body will attack them, which causes inflammation. And when it causes inflammation, it's diarrhea and upset. But luckily with regular E. coli, it doesn't result in death. This is what we hear a lot of times, and I'm glad you brought that up. A lot of times when we travel, you hear traveler's diarrhea, right? You drink water. You hear that in Mexico. If you go to Mexico, if you drink the water or eat food that's been rinsed with the water, you'll get sick. But then you look around and the locals are fine, right? And you're going, well, what the heck? It's because water and food can have different bacteria in it than the kind that you're used to eating. So let's say I drink water or eat food that has a little bit of Pseudomonas or E. coli in it. Now, normally here we have different strains and my body recognizes them, kills them right away. It's no big deal. If I travel somewhere though, they have different antigens. And so that makes my immune response more reactive. So now I get this intense diarrhea, I get upset stomach. But here's where it gets interesting. If you go to Mexico and you stay there for a month, you can start to drink the water because you get used to the bacteria. So if you slowly drink it, you actually get yourself used to the antigens and your body quits reacting. So it's just funny because it's a travel thing. It's something that we're being exposed to bacteria that are the same, they're the same genus and species, but just different surface antigens. So thank you for bringing that up. So other common foodborne pathogens then are ones that are going to be, like I said, the L157, and then also viruses. We do check for viruses. There was a hepatitis B outbreak recently at a Starbucks. Did you guys see that? There was, in the East Coast, they had a Starbucks employee who did not realize that he contracted hepatitis B. And he was preparing food and drinks and wasn't wearing gloves. And so they had to do a massive call out to anybody who went to the Starbucks on certain dates when that employee was working. And they had to give them free testing and then also vaccinations to try to make sure that they could keep their hepatitis B down. So hepatitis B is something that can affect the liver. It's usually self-limiting, but we don't want to cause any kind of long-term damage to our liver. So it's something that we're trying to get under control. So if we get exposed to an organism, one of the things that can happen is we can get infection. And infection is where the organisms are ingested, and then they cause illness, and then they are going to make you sick. And usually they're self-limiting. It's like around 4 to 5 days. You can also get what we refer to as intoxication, which means you're getting exposed to toxins. Now, we talked about endotoxins and exotoxins. So a lot of times I might have food that's sitting out and I have bacteria growing in it. Let's say I have chicken just sitting out. And then I'm like, oh, it's no big deal. It's been sitting out for 2 days, but I'm going to cook it. And when I cook it, I'm going to kill the bacteria. Okay, so you cook it and you kill the bacteria, but what have you left behind? The toxins. So for example, if I have chicken that has salmonella in it and I let it sit for a day, and then I'm like, it's good, I'm going to bake it. No worries, salmonella can't survive heat. I throw it in the oven and I bake it. It kills the salmonella, but salmonella produces endotoxins. So now I've released a whole bunch of endotoxins in that food. When I eat that food, I'm going to get a toxin poisoning or a toxin reaction. So even though there's not bacteria present, I'll still get sick. So this is why we have to be careful with our food handling. So one of the organisms I mentioned that can be dangerous is L157. We'll go through a few of our organisms here. It has an incubation time of about 2 to 4 days. And there was an event where we had some Cub Scouts that were camping that ended up getting exposed to L157 because they were making hamburger. They camped in and they brought some hamburger in their packs. And there was a couple of Cub Scouts that were helping make dinner that night, a couple of the young boys. And they were making hamburger patties, and one of them said that the hamburger patty that he made, he kind of put it to the side on the grill, and it didn't cook all the way through, and he took a bite of it later, and it was cold. And since it was cold and hadn't cooked, he ended up getting himself exposed to L157. Now, of course, this is a kid. They're like 10, so they didn't think about it. They're like, well, we eat cold burgers the next day, so they just figured, no big deal. But unfortunately, they were basically eating raw hamburger meat. And it was 2 kids that had that happen. They're kids, they don't know. And so they ended up getting exposed to L157. Both of them survived, but during the next few days of their camping trip, they started having upset stomach and not feeling well. They went home, they still had upset stomach, they started having diarrhea. Their parents saw that they had what's called the hemolytic uremic syndrome, which means that they're getting bloody stool. And they took them to the hospital right away. And luckily they were treated and they were fine. But it just took a while to figure out where they got exposed. It's like they had to be interrogated to try to figure out where they got exposed to these things. Yeah?
How does kidney failure result from that?
Good question. So, E. coli L157 starts to grow inside of the intestine. It starts releasing toxins as it dies off. Those toxins will start going through the kidneys as a filtration, and the toxins are what actually start to cause damage to the kidneys. Now what's going to happen is the kidneys start to have inflammation, and our immune system over-responds. Our immune system starts attacking the toxins, and in turn, a lot of times it will start attacking the kidneys. Then our organisms, our body and our organs start going into stress mode, and they start doing a systemic shutdown. We start shunting the blood to the heart and the brain, trying to protect it. We pull blood from all the other organs, and so the other organs start to have suffering from dying of not having enough blood coming through. So it's kind of an overreaction that we run into with coli 157. Oops. The next one is salmonella. Salmonella is one that we mentioned with typhoid Mary. So again, we have a lot of different strains of salmonella out there. The last count, we had about 2,000 strains, meaning ones with 2,000 different types of antigens. There's about 10 of them that are really associated with food illness. And usually where we hear about salmonella is when you eat like raw cookie dough or eggs, right? Now, again, if you're collecting fresh eggs, you don't have to worry about it because they have bloom around them, right? But usually it's when you're purchasing store-bought eggs that have been washed and then later exposed to fecal matter and they've lost that protective bloom. And so now the salmonella, in theory, can leach through the eggshell and get into the yolk. And that's why they tell you to make sure that you cook and don't eat raw eggs. I don't know. My family, we've eaten raw eggs. Raw cookie dough. Every year for Christmas, we bake a bunch of cookies. And my kids eat it as faster than I can make it, kind of a thing. I'm like, you're gonna get salmonella, and so far they're all still alive. So I don't know. It's like they're always fine. So I think it's one of those things where it doesn't occur very often, but when it does, it's miserable. So again, salmonella would basically be self-limiting. You have diarrhea, nausea, fever. You don't have to be hospitalized. They're not going to give you an antibiotic. Because if you do, they're just going to kill the salmonella too fast and have too much endotoxin get released. So they just let it run its course. And your immune system will wipe it out, but it's just going to be about 4 or 5 days of misery. Listeria, again, this is the one we worry about for pregnancy. It has a much slower incubation time, anywhere from 2 days to 3 weeks. Listeria a lot of times is associated with meat and seafood. That has been in contaminated conditions, meaning that they haven't been stored properly, or they haven't been properly sanitized, or with milk that hasn't been pasteurized. We had a listeria outbreak with cantaloupe a few years ago in Santa Maria, and it was actually nationwide, so it was pretty bad. What happened was the cantaloupe that was being harvested was being run through a machine that was processing something. I was trying to remember what else it was processing. I think it was sprouts. So they ended up with E. coli and they ended up with listeria in this machine because they weren't cleaning it properly. So they'd run the sprouts, and it's always sprouts. They run the sprouts and then they run the cantaloupe. And it was putting bacteria on the outer edge of the cantaloupe. Okay, well that's not too bad. But when you would sell it and people would get their knife and cut through the cantaloupe, they cut through the rind and then pull the bacteria through the melon part of the cantaloupe. So now if they consumed it, they were getting higher amounts of bacteria. So that was how they were getting exposed to that. So how can we prevent that? You can wash your cantaloupe. That's weird. We don't think about washing cantaloupe and watermelon, but it's something that we probably should do. Especially with the listeria outbreaks. It's just a good idea. You can do an acid rinse, like with vinegar or with Clorox just on the outside really fast. And a diluted Clorox, 10%. Campylobacter, as I mentioned, you can have bloody diarrhea, nausea, fever. This is one that we usually again see with contaminated meat products, again, like pigs or cattle that have been in unsanitary conditions. Sometimes we'll see it get into water with fecal matter from cows or pigs. Again, it's self-limiting. It's usually about 2 to 5 days of just having intense diarrhea, and then it just kind of goes away. If you've ever had food poisoning, that's what all of these feel like. So these are easy to study because all of their symptoms are the same, right? Staph aureus. So staph is always a concern because it doesn't require as many cells. You only need about 100,000 colony-forming units, which isn't a whole lot. We have that on our plates every time we plate staph. It also has a very fast incubation. If you've ever gotten food poisoning right after eating something, so for example, you had some dinner and then within a few hours you start feeling really sick and you're pretty sure that's where you got it from, it's going to be staph because staph is the one that gives you a fast reaction. All the others are going to be a slower incubation, but this one is the one that is a quick one. And usually you're not responding to the staph, you're responding to their exotoxins. So their exotoxins are usually the ones that are there. So a lot of times you hear about like potato salad poisoning, like mayonnaise, things like that. That would be your staph. Now what's funny is the American Society of Microbiology, they were trying to figure out what it is about like potato salad in particular or mayonnaise that causes bacteria to grow. And they did this study where they put like a variety of potato salads out at different temperatures in the sun and the, the shade. They tried this whole big study. It was like basically a picnic at ASM, and they were trying to poison people. I mean, they didn't have people eating it; they were sampling it, and they never could get a high enough level of staph to cause illness. So what they finally realized is it's not like the mayonnaise is bad or the potato salad is bad. It's like a whole a sequence of events where a bunch of hurdles have been broken. So maybe the mayonnaise was stored incorrectly, maybe the mustard was stored incorrectly, maybe the potatoes were stored incorrectly, something along those where we had multiple events. And then it's just like this, chaos that occurs. And that's how we end up getting our potato salad that goes bad. It's like we have to have several hurdles that have fallen for us to actually get sick. So you don't have to always have this fear, I guess is what I'm telling you. The chances of something happening are pretty low. Yeah.
You said that we usually don't get sick from staph, we get sick from their exotoxins.
Correct. Yes.
Earlier, did you say endotoxins are a different one?
Remember, endotoxins are part of the LPS layer, so those are the gram negatives. So salmonella has endotoxins, they're in the cell wall. And the only way they get released is if the cell dies. Exotoxins are produced as the cell is growing. We usually see it with gram-positives, and they spit them out as waste product. So I can get exotoxins released while the cell is still alive. Endotoxins get released when the cell dies. You got it. CBA, again, we mentioned this one before. This is one that we always worry about for meat products that haven't been stored properly. Again, usually if you get exposed to CBA, you start to have nerve impulse responses where you start to feel dizzy, you start to feel like your tongue is going numb, you have trouble breathing and swallowing. Anytime you have trouble breathing or swallowing, please get to the ER ASAP. It's not something that you want to mess around with. Even if it's not CBOT, if you're having any kind of those difficulties, you definitely want to get there and get that checked out. And then also, these are ones that can grow in foods that have a pH of greater than 4. So they can grow in slightly acidic foods, and they can grow in low oxygen foods because of the fact that they're anaerobes. So how can we prevent foodborne illnesses? Well, basically make sure that we're storing our meat properly. And then once we cook our meat or our eggs or whatever we're cooking, make sure that they're getting to the proper temperature. So you want to have it at least to 160 degrees Fahrenheit. We want to also make sure that we've stored it correctly so that we don't have a lot of growth that's occurred with toxins. So that's why, you want to keep your turkeys refrigerated until you're ready to cook. You thaw your turkey in the fridge because you don't want bacteria to grow. So things along those ideas. Avoid cross-contamination. If I am cutting turkey, I don't want to, or chicken, I don't want to immediately start cutting tomatoes or fruits on that same cutting board. I want to use different cutting boards for my food. Keeping my utensils clean, my work area is separate, so that way I don't have, I'm not making raw hamburger here and right beside me somebody's cutting up peppers. That's just too close. We can have cross-contamination occur. And then just, staying vigilant in your kitchen, making sure you're keeping things clean. Always keeping foods cold. Remember, coldness isn't going to kill bacteria, it just slows them down. So we're trying to keep our foods cold so that we don't have a huge influx of growth. We could still have spores present, but the idea is that if you keep them cold, the spores won't hatch. And so that's what we're trying to do is keep the spore counts down. And then washing our fruit and veggies. So again, you can do like an apple cider vinegar rinse. You can do a white vinegar rinse. You can do a 10% bleach solution rinse. I just have a spray bottle at my counter and I just quickly spray my fruit when it comes in and then I wash it with water again. So that way it doesn't taste like bleach. It doesn't taste like anything, but I know it's clean. And again, of course, my kids eat the grapes on the way home, so it doesn't matter anyway. But at least it makes me feel like I've tried. I don't know. It's like I don't know how any of us survive. Do you ever see this stuff and go, "How are we still here?" Right? Yeah. So obviously the risk is not horrid. I don't want you to have like these fears, But it just cracks me up because everything we do that's like careful. We look over at our kids, we're like undoing it. It's like whatever. Okay, fine, we're still alive. So bacteria, illnesses are still pretty rare and pretty low. So we're pretty good. Part of that is because a lot of our food is acidic. A lot of our food does have preservatives that keep the growth down. So they've got our backs. And then personal hygiene. Of course, please wash your hands when you're cooking food. You don't have to ruin your hands. If you're working in a restaurant, you need to wear a hairnet to keep your hair out of your food. keep plastic gloves on your hands so that you don't spread anything. And then one of the big one is if you are preparing food or if you're a waitress or a waiter, if you're not feeling good, then don't touch the food that day. Stay home that day. And that's really hard to do. Thanks, Erin. Wait till Erin gets that. They apparently have like a migrating group of students. Thank you, Erin. It's like the student migration is occurring. It's like, okay. So if you're sick, try to take that day off. And I know that that's really hard because a lot of times if you take a day off, you lose pay. So we have people that are, kind of in a situation where they have to go to work. If you do, make sure you wear gloves, make sure you wear a mask, do everything you can to make sure that you're not spreading the illness. And with that, we are done. So I'm going to hand back your quizzes. We'll go over your quizzes, and then that's it. Y'all are free.